Everyone, I'm Tricia Thompson from GlutenFree Watchdog. Last week I had the pleasure of speaking at the Food and Nutrition Conference and Expo, which is held annually by the Academy of Nutrition and Dietetics. I spoke alongside Carol DeLima of the FDA. Carol's presentation was about FDA's proposed rule for gluten-free labeling of fermented and hydrolyzed foods. I spoke about testing fermented and hydrolyzed foods for gluten and the difficulties involved with doing this. This video is meant to be watched alongside the PowerPoint presentation. I've posted this PowerPoint presentation in the same area as this video. There are 6 slides per page. Hopefully you can see them. So please follow along with the PowerPoint as you listened to me walk you through these slides. And just a word of warning, this video will probably last about 20 to 30 minutes. So if you have that printout, and again, I'm going to be looking off to the side because I'm actually looking at my slides and I don't have 2 computers set up. So if you go to the slide that's titled Testing Food for Hydrolyzed Gluten and Why This is So Difficult, and then looking at the next slide, as I said to dietitians at the presentation, at the end of this presentation, you should be able to state the differences between the 2 main types of ELISAs that we use to test food for gluten. You also should be able to discuss some of the limitations associated with testing foods containing hydrolyzed gluten, and you should be able to name some of the hydrolyzed foods and ingredients that are difficult to accurately assess for gluten. And knowing this information, having it at your fingertips, will really help you when you're contacting manufacturers with all of the questions that you have. So the first thing that we're going to talk about is testing that we specifically use to assess food for gluten. And we use what's called an enzyme-linked immunosorbent assay, which is otherwise known as an ELISA. And this is what is commonly used to assess food for gluten. As some of you may know, this assay involves an antibody-antigen reaction. So what this means is that antibodies in the assay will react with an antigen if it is actually present in the food sample. So the antigen that we're concerned about at this time is gluten. There are 2 types of ELISAs that we currently use to assess food for gluten. These ELISAs are the sandwich ELISA and the competitive ELISA. And the main difference between the 2 is that the sandwich ELISA requires 2 epitopes or antibody binding sites, and the competitive ELISA requires only 1. And if you need a trick to remember, just think of a sandwich with 2 slices of bread, 2 slices of bread, 2 epitopes. So let's talk a little bit specifically about the sandwich ELISA that is most frequently used to test foods for gluten. This is the sandwich R5 ELISA. This is not the only sandwich ELISA that's available for testing food for gluten. It just happens to be the most frequently used worldwide. It's based on the R5 monoclonal antibody to the 5-amino acid sequence QQPFP, and this sequence repeats frequently in gluten protein. It is the Codex Alimentarius type 1 method, and it is used to detect and quantify proteins from wheat, barley, and rye that remain relatively intact, meaning that they have not been broken down into smaller protein fragments. The competitive R5 ELISA is a modification of the sandwich R5 ELISA, and this is the ELISA that we use to test food for the presence of hydrolyzed gluten, gluten that has been broken down into smaller protein fragments. So one of the questions that I am asked fairly frequently is why can't we use the sandwich R5 ELISA to accurately assess food for for gluten when it may contain hydrolyzed gluten protein fragments? And the answer is that hydrolysis will break the gluten protein into varying lengths of protein fragments. And it may in fact be the case that the fragments are too small to contain those 2 QQ-PFP epitopes that we need in order for the sandwich R5 ELISA to work, to actually be able to detect those protein fragments. So for an example, if you look at the sequence of amino acids that is represented on the slide, and you see that it has 6 repeating QQ-PFP epitopes. The little a's just stand for other amino acids. And also keep in mind, this is important, that QQ-PFP stands for glutamine, glutamine, proline, phenylalanine, proline. And those prolines become very important when we talk about beer in a few slides. So if we hydrolyze this particular protein, we conceivably could end up with the 4 fragments that are represented on the next slide. And as you can see, fragments 1 and 4 contain 2 QQ-PFP epitopes. These fragments would be detected by a sandwich ELISA, but fragments 2 and 3, which contain only 1 QQ-PFP epitope, would not be detected by the sandwich. They would in fact be detected though by the competitive R5 ELISA. Unfortunately, the competitive R5 ELISA is not universally accepted by all regulatory agencies. So for example, it is approved by the Association of European Celiac Societies, as well as the American Association of Cereal Chemists International, but it is not accepted by the FDA and the TTB, and questions have also been raised by some testing experts. One of the questions that is frequently asked is whether this assay is more qualitative than quantitative. In other words, the competitive can be used to detect those gluten protein fragments, but at what point do the number of fragments become harmful? And can you actually take gluten protein fragments and translate those fragments into a part per million level of intact gluten protein? So the FDA actually questions this in their proposed rule for gluten-free labeling of fermented and hydrolyzed foods. And they write, there is uncertainty about whether these methods can quantify the amount of protein from which those fragments were generated by hydrolysis. This uncertainty creates problems in equating these test results to an equivalent amount of intact gluten in the fermented or hydrolyzed product. Another question that's frequently raised is whether the R5 epitope is found in all toxic sequences. So it's important to understand that the R5 epitope itself is not toxic. It is simply a marker protein for longer sequences of amino acids that just happen to contain the QQ-PFP epitope. So it might be the case that the R5 epitope occurs in a fragment that is not toxic, while it does not occur in a sequence that is toxic. So let's look now at some of the difficult to test products. These products include barley malt and barley malt ingredients such as malt extract and malt syrup, as well as beer and probiotics. And the reason that these are all difficult to assess for hydrolyzed gluten is because, in fact, they contain hydrolyzed gluten, and you cannot simply assess them for gluten contamination using the sandwich ELISA. This is not a complete list by any means. These are simply some of the products that we receive the most questions about at GlutenFree Watchdog. So I want to talk first about malt ingredients. The bottom line on malt ingredients is that malt, malt extract, malt syrup, and malt vinegar cannot be included in foods labeled gluten-free and sold in the US. But as those of you who are members of GlutenFree Watchdog know, this does not prevent many products from actually being sold in the US labeled gluten-free and containing these ingredients. This is being done wrongly, obviously. This should not be the case with these products, but we're seeing it happen at an increasing rate, especially for me. I mean, I'm dealing with these manufacturers, it seems, every week. This is happening with both imported and domestic products, although I would say it's happening more frequently with imported products. And this is probably because some malt ingredients are allowed in foods labeled gluten-free in the UK and the EU, depending upon the gluten levels in the final product. So this is a difference between gluten-free labeling law in the United States and outside the United States. As far as definitions of malt ingredients, many of you already know this, but it's important to stress again. Malt is an enzyme preparation obtained from barley, and malt extract and malt syrup are both water extracts of germinated barley grain. The common or usual name for barley malt is malt, and the common or usual name for barley malt extract is malt extract. And the reason that it's important to know this is that if you see the ingredient malt on a food label, it means barley malt. And if you see malt extract, it means barley malt extract. And again, the reason that these malt ingredients cannot be assessed for gluten using the sandwich R5 ELISA is that they are naturally hydrolyzed by enzymes that are present in barley grain. So that barley protein will be broken apart into smaller protein fragments, and these fragments may or may not contain 2 QQ-PFP epitopes. So you have to use the competitive R5 ELISA on these products or another competitive ELISA. Malt vinegar is also a barley-based product, and it also cannot be assessed for gluten using a sandwich ELISA because it is a fermented product, and again, that fermentation will break that barley protein down into smaller protein fragments. The FDA has clarified their position on the use of malt ingredients in food labeled gluten-free. They did this during an Academy webinar sponsored by the Medical Nutrition Practice Group. What I'm about to read to you is considered FDA's current view on malt ingredients. They actually confirmed this at the session at FNCE. So they stated during the webinar, malt extract and malt syrup are derived from gluten-containing grains and containing gluten. Therefore, we consider them as ingredients not processed to remove gluten, and they would not be permitted in foods bearing the gluten-free claim. They went on to state, if malt syrup or malt extract are further processed in some way to remove proteins, we believe that would change its nature to such an extent and alter the characteristics of the ingredient so that they would no longer bear the same common or usual name. In other words, they would not be malt, and they would not be malt extract, and they would not be malt syrup. FDA also clarified their stance on malt vinegar in the proposed rule, writing, some malt vinegars are the product of fermentation without distillation of an infusion of barley malt or cereals whose starch has been converted to malt. Because these types of malt vinegar are derived from gluten-containing grains that have not been distilled or otherwise processed to remove gluten, They may not be used as ingredients in a food bearing a gluten-free claim or bear such a claim themselves. Unfortunately, as I stated, regardless of these rules and regardless of the fact that malt, malt extract, malt syrup, and malt vinegar are not allowed in foods labeled gluten-free, to date, in 2016, GlutenFree Watchdog, meaning me, I have personally dealt now with 13 manufacturers. The slide says 12, but I was dealing with one right up until I left FNCE. 13 manufacturers of foods that are labeled gluten-free yet contain malt ingredients. I've spoken to every single one of these manufacturers, and they all believe that they are in compliance with the FDA's gluten-free labeling rule because their products are testing under 20 parts per million using the sandwich R5 ELISA, and in some cases they are using the competitive. They are under the impression that if their product tests below 20 parts per million, then almost by definition, any ingredient in that product must in fact be processed to remove gluten. This is not the case. This is not what the FDA gluten-free labeling rule means. The next slide is just an example of one of the products that we dealt with at GlutenFree Watchdog, and as you can see, the ingredient list includes gluten-free barley malt. So remember, this was a presentation to dietitians. So this slide says, how should dietitians talk with consumers, their clients, about these issues, about malt and gluten-free foods. And so what I want to tell you is that we really want consumers to trust the gluten-free label, but if you do come across a food that includes malt, malt extract, malt syrup, malt vinegar in the ingredients list, and it is labeled gluten-free, you should avoid it. And you should avoid it even if you contact the manufacturer and they tell you that, oh, their product tests below 20 parts per million, or those ingredients have been processed to remove gluten. You also may want to consider contacting your local FDA consumer safety officer. They're very pleasant to deal with, and they will listen to you, and you can find a state-by-state regional listing at the link on the slide. Okay, so let's talk a little bit about beer. And as most of you know, this is very controversial right now in the gluten-free community. The bottom line on beer is that it's regulated by either the TTB or the FDA, depending upon whether the definition of a malt beverage is met. So beers that are regulated by the TTB meet the definition of a malt beverage, and they cannot be labeled gluten-free. They can be labeled processed to remove gluten, and we're going to talk more about that in a couple slides. Beers regulated by the FDA are not considered malt beverages, and under FDA rules, they may be labeled gluten-free if all criteria of the gluten-free labeling rule are met. Beers regulated by the TTB and labeled processed to remove gluten are sometimes, unfortunately, represented as gluten-free on restaurant menus, in bar menus, on shelf tags in grocery stores and package stores, in other words, liquor stores. So as far as definitions of malt beverages, a malt beverage is actually a beverage that contains both malted barley and hops. So beers regulated by the TTB, by definition, contain both malted barley and hops. Beer cannot be assessed for gluten using a sandwich ELISA because it is a fermented beverage. So the barley protein, the malt, will be broken down into smaller protein fragments. Beer regulated by the FDA is not made from both malted barley and hops, so it is not considered a true malt beverage. Beers regulated by the FDA are made from either hops and a substitute for malted barley, or malted barley but not hops. But at this point, I am not aware of any beers that are made from malted barley and not hops. Some may exist, and if you come across one, please let me know. So as far as beer and TTB rules and gluten-free labeling, again, beer regulated by the TTB cannot be labeled gluten-free. It can include the claim processed to remove gluten if in fact it has been processed to remove all or some of the gluten, and it also includes another statement on product packaging which reads, product fermented from grains containing gluten and processed to remove gluten. The gluten content of this product cannot be verified, and this product may contain gluten. Two examples of popular gluten-removed beer are Omission and Dora. As far as FDA rules, gluten-free labeling in beer, as I stated, beer regulated by the FDA can be labeled gluten-free if it is made from a gluten-free substitute for malted barley. Beer regulated by the FDA and made from a gluten-containing grain cannot be labeled gluten-free unless, and quoting from the proposed rule, and this is confusing, the gluten-containing grain has been processed to remove gluten in accordance with the provisions in the gluten-free definition prior to making beer. But the FDA states in the proposed rule, we are not aware of any scientifically valid way to evaluate such a claim. So at this point, beers regulated by the FDA and made from a gluten-containing grain do not appear to be able to be labeled gluten-free. It seems that the FDA is leaving open a window in case something changes, but hopefully they will clarify this in the final gluten-free labeling rule, because this is quite a confusing statement, even for those of us who follow what's going on with the FDA. So as far as testing beer for gluten, this is where things get really tricky, and this is where I really want you to understand what I'm saying, because this impacts that Omission beer, which some of you may drink, or you may have friends who, and you may see Omission beer talked about a lot in social media. There are additional limitations to testing beer over and above what we see with other fermented products. Gluten-removed beers may be treated with what are known as prolidase enzymes, such as brewer's Clarex. Prolidase enzymes break apart protein at the amino acid proline. Now remember that I told you that QQ PFP epitope, the P's stand for proline. So if the prolidase enzyme breaks apart the QQ PFP epitope, you no longer have that epitope available that allows the competitive and the sandwich R5 ELISA to work. So of course, running the competitive R5 ELISA or the sandwich R5 ELISA on these beers is not going to find anything, because in the opinion of many, many experts, this makes the competitive R5 ELISA useless in measuring the gluten content of beers treated with prolidase enzymes. So the FDA actually addresses this issue in their proposed rule, writing, adding these haze control enzymes may generate peptides that are not detectable using the commercially available competitive ELISA-based methods that rely on the presence of proline in the epitopes. However, it is uncertain that cleavage at proline residues totally eliminates the concern for people with celiac disease, because there may be immunopathogenic protein fragments still present. Regardless of testing constraints and TTB labeling rules, it is not uncommon for beers labeled as processed to remove gluten to be listed as gluten-free on restaurant menus. This is a photograph of my husband, and he is pointing at a menu blackboard, and as you can hopefully see, omission is written on this blackboard, and it is represented as gluten-free. So what is my advice, what was my advice to dietitians as far as what to tell all of you about beer and gluten-free labeling? It's my very strong opinion that at this time you should avoid gluten-removed beers. It's also the case that you may not be able to trust the gluten-free designation on restaurant menus and bar menus, as well as shelf tags in grocery stores and liquor stores. So it's important to actually look at the gluten-free label. Now, this is easy to do in a grocery store or a liquor store. It's next to impossible in a restaurant or a bar. So it may be helpful for you to familiarize yourself with some of the more common gluten-free beers, those regulated by the FDA, and gluten-removed beers, those regulated by the TTB. At the very least, know that omission beer is a gluten-removed beer. Regardless of what you see on a restaurant menu or on a bar menu, nothing has changed. It remains gluten-removed. The last product category that I want to talk about are probiotics. Probiotics may contain bacterial strains grown on gluten-containing fermentation media. It is unclear at this time whether use of a gluten-containing fermentation media will disqualify a product from being labeled gluten-free. And as I mentioned during the conference presentation, it's really hoped that the FDA addresses this issue in their final rule. As far as regulations of probiotics, they're generally regulated as supplements. So what this means is if a probiotic is labeled gluten-free, it must be in compliance with FDA's gluten-free labeling rule. An ingredients list is required on the label of all supplements. So for probiotics, this means that the bacterial strains are generally included. The growth medium is generally not included in the ingredients list. I've never seen it. That doesn't mean it doesn't exist on some probiotic, but I haven't seen it. And the reason that probiotics cannot be assessed for gluten using a sandwich ELISA alone is that residual gluten from the fermentation media may be in the form of gluten peptide fragments. So one of the reasons why I'm concerned about probiotics and residual gluten has to do with some of the testing that we've been doing at GlutenFree Watchdog. As many of you know, we've tested a number of probiotics. One manufacturer of a probiotic that was labeled gluten-free was using a barley-containing growth medium in one of the bacterial strains. The specific growth medium was yeast protein from spent brewer's yeast. For those of you who are not familiar with spent brewer's yeast, it is a byproduct of the beer brewing process, so it will be contaminated with malt. When we tested this particular probiotic, it tested well above 20 parts per million of gluten using the competitive R5 ELISA. When the manufacturer changed the growth medium and went back to using a bacterial strain that was grown on molasses, this new formulation tested well, I shouldn't say well below, it tested below 10 parts per million using the competitive R5 ELISA. 10 parts per million is the lower limit of quantification, so that's as low as we can go with the competitive as far as being able to quantify. So what did I tell dietitians to tell consumers? I advise dietitians to tell all of you that you really should only use those probiotics that are labeled gluten-free. If you're concerned about the growth medium, please contact the manufacturer and ask. I don't know whether they will tell you. They may tell you that they do use a gluten-containing growth medium, that they test their product, and their product tests below 20 parts per million of gluten. I will tell you that probiotics are really tricky to test for gluten, and it is not enough to test with the sandwich because of the potential for the hydrolyzed gluten protein fragments. So if you ask them what test they're using and they don't know, or they tell you the sandwich, just know that that is not enough, and they really need to use the competitive. So to briefly summarize what I've been talking about for the past however many minutes, sandwich ELISAs require 2 epitopes and competitive ELISAs require only 1. Gluten protein that has been hydrolyzed may not be detected by a sandwich ELISA, and ingredients that cannot be accurately assessed for gluten using only a sandwich ELISA include malt, beer, and probiotics. Hopefully this has helped clear up some of your confusion. Hopefully it hasn't made you more confused. If you have any questions, please post a comment. This is the easiest way for me to make sure that I reach the widest audience, and it prevents me from having to repeat myself. But please do feel free to post your comments. Thank you so much. I know this was long, but hopefully it was helpful. Bye.